Hello, in this video we'll see some of the enzymes involved in recombinant DNA technology, that is terminal transferase, lambda exonuclease, polynucleotide kinase, alkaline phosphatase and S1 nuclease. Terminal transferase is also known as terminal deoxynucleotide transferase or DNA nucleotide exotransferase. It is a DNA polymerase purified from calf thymus. It adds oligodeoxynucleotide tails to the 3' ends of DNA duplex or single-strand DNA. It is helping in homopolymer tailing. It doesn't require any primer as the case of DNA polymerase, and it prefers 3' protruding end created by a lambda exonuclease or restriction endonuclease like PST1. In this figure, oligo G tails are added to the 3' end of the plasmid DNA and oligo C tails are added to the 3' end of the foreign DNA to create a recombinant plasmid by homopolymer tailing. Uses of terminal transferase: for labeling the 3' end of DNA, adding complementary homopolymer tails, and to clone cDNAs into plasmid. Lambda exonuclease is an enzyme that selectively digests the 5' phosphorylated strand of double-stranded DNA. The preferred substrate is a blunt-ended 5' phosphorylated double-stranded DNA. It creates an improved substrate for terminal transferase. In the figure below, the blunt-ended DNA is digested by lambda exonuclease to create a 3' overhang and removing 4 of the nucleotides. The next enzyme is kinase. It is taken from T4 bacteriophage, also known as polynucleotide kinase, is an enzyme that catalyzes the transfer of the end phosphate of ATP to the 5' end of either DNA or RNA. It is used in radiolabeling 5' end for sequencing by Maxim-Gilbert method, for terminal labeling, and to phosphorylate synthetic linkers for ligation. In this figure, the 5' end of the DNA is phosphorylated by polynucleotide kinase by transferring the gamma phosphate of ATP. Alkaline phosphatase is an enzyme that removes the 5' phosphate group from DNA. It is isolated from bacteria and calf intestine. It is used to prevent recircularization of plasmid and intramolecular association, or to prevent the dimer formation, that is intermolecular association. The action of alkaline phosphatase is opposite to that of kinase. While kinase adds the phosphate group to the 5' end, alkaline phosphatase removes the phosphate group from the 5' end. The enzyme S1 nuclease is isolated from Aspergillus oryza. It cleaves a single-strand DNA, leaving very short single-strand gaps within the duplex DNA. That is, 1 or 2 bases gaps are created. It cleaves supercoiled DNA having single-strand bubbles. Uses: to distinguish non-supercoiled and nicked DNA circles from supercoiled DNA. Individual regions of a gene can be isolated. Can be used to produce RNA-DNA hybrid with a gap in DNA strand, and we can remove the flanking regions and the introns. On alkali treatment, the RNA is removed off and we can isolate the DNA fragments, thereby identifying the exons. It is also used for mapping of DNA.